He is trained in synthetic biology and genomics, earning a PhD from UNC in the field of epigenetics. Currently, he leads the lab automation and bioinformatics teams at Locus Biosciences, an engineered bacteriophage company developing solutions for antibiotic-resistant bacteria and microbiome dysbiosis. Give it up for Dr. Taylor Penke.
All right, I'm excited to be a part of the event. I'm here to talk to you tonight about the silent pandemic, or the increasing rates of antibiotic resistance that are occurring in society today. So antibiotics have been some of the most effective and successful drugs in the history of pharmaceutical development. They've been around for almost 100 years, dating back to the discovery of penicillin in 1928. But over time, their efficacy is decreasing as antibiotic rates or antibiotic resistant rates increase. And we also know that antibiotics oftentimes kill your good bacteria as they're taking out the bad bacteria, the target pathogen that you're trying to eliminate in an infection, which really leads us to question the paradigm of how we're treating infections today in society. So at Locus, the company I work at, what we're working on is a platform to reach into the human body, eliminate the target pathogen very specifically while leaving the rest of the human microbiome or a patient's microbiome intact. And to do that, we're leveraging the bacteriophage or the phage, which is the most abundant organism on Earth. It's all over this room, for example, and they're simply viruses that target bacteria very specifically. They work like many other types of viruses. They bind to the surface of a bacterium cell, inject their DNA, and then make more copies of themselves, eventually rupturing the host cell and killing the bacterium. So although they may look like something from Stranger Things, we think that these are possibly one of the most potentially positive tools that we could use for human health. But their specificity and the vast diversity that exists on Earth makes them really challenging to work with. So in this heat map that I'm showing on the left here, the x-axis is a number of different types of E. coli, different E. coli strains. And on the y-axis are a number of different phages. And the heat map is colored so that darker colors indicate better activity of a given phage. It's better at killing that bacteria. And there are two things that I hope you can appreciate from this graph. One is that phages have totally different patterns of activity of which types of bacteria they can kill, and also that you would need a combination or a cocktail of phages to be able to kill all of the E. coli strains on this particular map. And choosing that optimal combination of phages for your cocktail is one of the most challenging aspects of working with them. And so if I can take you back to middle school math for a second, the question that I always hated on tests was, you have 10 different colored balls, you put them all in a bag. How many different combinations of 3 colored balls can you pull out of that bag? Well, if you do that with phages, say you have 250 phages that you put in that bag, and you want to assess all potential 6 phage combinations that you can use, that's 100 billion different combinations that you would need to test or assess in some way. So these types of challenging problems really lend themselves to the intersection of automation and AI, which is why we're all here tonight. So we set out with a thesis that to build a good drug product, we would need to discover thousands of different bacteriophage and test them on thousands of different bacteria. And to do that, we decided to develop an automated platform that, for the purposes of this talk, I'm boiling down to 3 main capabilities. One is storage and retrieval. If you have thousands of different biospecimens, you need a way to access them in a traceable way and organize them effectively. And you also need some way to assess how good your phage or your phage cocktail is on all these bacteria, which involves mixing different liquids together and having some measurement device. And then finally, even the best of automation would struggle to test, at least in an efficient way, 100 billion different combinations. So we think that there's a strong role for leveraging AI to predict those right combinations of phages. So we set out to establish a storage and retrieval capability. We worked with a group called Hamilton Storage Technologies to develop these automated biobanks, which are essentially vending machines for biospecimens. We wanted to make getting 100 phage or 1,000 phage out as easy as getting a Coke for our scientists, or maybe even a little easier because these don't take your money and you don't have to take a dollar bill and rub it on the side of the instrument to get your particular sample. And so how these work are all your specimens are in these tiny little tubes that have barcodes on the bottom of them. They're these green capped items here. And this biobank has a robotic arm that can move these different racks into position, and a tube picking device that allows you to select the right specimen that you want and organize them into an appropriate pattern for a scientist to use in their downstream application. And when you have all those organized, you can use this automated capper-decapper device to remove these caps, making it really easy to put these types of samples into downstream robotics. So with all of your samples in place, you need a way to actually mix the phage and bacteria and assess them. And to do this, we use a couple of work cells that are liquid handlers, which they're boiled down to their basic function. They're automated liquid movers. They have channels that can automatically aspirate and dispense liquids and move them together into these plates that have hundreds of different individual reaction containers. And many of them have robotic arms like ours here to move these plates into different measurement devices so that you can scale to a high throughput assessment of your phage. So essentially the question that we're trying to ask is which phage combinations have the best antibacterial efficacy on different strains. And we follow this simple workflow, which is essentially mix your phage and bacteria, allow the phage time to kill that bacteria, and then spot the remaining bacteria onto a media surface where they can grow into a colony, image that plate, and then simply count how many bacteria are left and how much has the phage been able to kill. So this video is an example of the automated imaging method that we use. This method scans barcodes on plates, and then in a second you'll see a slow motion video of the arm placing a plate onto a light surface to take a quick picture. And what you get from this are images like the bottom here, where you have 96 different spots, and you can simply count those bacteria colonies that are present, and it's pretty easy to pick out which phages have been the best in those blank spots that are in the middle of the image. To do this at scale, we developed a computer vision algorithm that can automatically count all of those colonies and all those reactions, and we've used that to build a database of around 4 million different combinations of phage cocktails and bacteria interactions, which obviously large databases are great for leveraging AI. And we're in the beginnings of our story here with leveraging large language models, but we see a lot of promise in using this for our scientists to be able to access these databases and get recommendations from these models. So this example, the user is asking for a recommendation for a cocktail on a particular bacteria strain. This model accesses a database on the back end, gives a recommendation, and also a rationale for why it decided to choose that. And like all large language models, you can interface it more directly and ask what are the phages that are present in that sample, so a scientist can then go into the lab and make that prediction or test that prediction. So while we're really excited about this platform and what we can use it for, we're even more excited to bring this into the clinic. And we're in the middle of a phase 2 trial right now for patients with recurrent urinary tract infections. So these are patients that are often have multi-drug resistant bacteria. They're coming back to the clinic for more antibiotics every 3 or 6 months. And so this particular drug targets E. coli. It's dosed through a catheter as well as IV. And in this trial, we're co-treating with a standard of care antibiotic. We've just completed part 1, which is unblinded, non-placebo controlled, a smaller patient set. But we're really excited about the results that we've been seeing. The middle panel is essentially measuring how much phage is present in urine, and as we expected, we have really high levels of phage present. And what we hope for and what we're really excited about is the dramatic reduction of bacterial burden, which is shown in the far right graph there. On average, we're seeing about a 1,000,000-fold reduction of E. coli levels in these patients. So we're really excited to take this into the placebo-controlled portion of the trial and look at statistical numbers to see how effective this drug product may be. So the thing that I want to leave you with today is hope for tools that we and others are building to turn what's been a burnt earth approach to treating infections for the last 100 years to something that's much more like a precision targeted strike. So happy to take any questions. Thank you.
Thank you. Dr. Penke, I'm over here to your right. Dr. Penke. I have a question right here.
Thank you for your presentation. I was wondering about how hard was it to implement these highly automated processes, and what were the struggles that you went through to succeed in implementation?
Yeah, lots of struggles. I'll say the ones that come to mind most are really effectively testing different quality control measures. So when you're working with tons of bacteria and phages are known to They don't actually jump, but that's the joke, that they get all over all of your samples or into neighboring wells. And so the types of quality controls and the different types of labware testing that we needed to put in place to be able to make sure that contamination wasn't occurring into neighboring wells and make sure that the results were essentially reliable was one big challenge. And then also just setting up these types of automated systems. There's so many different things that can go wrong. The more complex you build your system, the more things that can go wrong. And so over time, just continually optimizing to reduce those errors, the frequency of them, and then putting in automated checks to recover from those errors that are most common. Those are a couple of the biggest struggles that we dealt with.
Other questions? Go right here.
Thank you. How long did it take? How much did it cost?
Excellent questions. So we've been at this as a company for 9 years. We've been developing our automation for 5 years. We actually acquired a company from San Francisco that had the initial liquid handler that was our gateway robot into the space. Ultimately, between labor and capital equipment, we've invested probably $2 to $3 million into these particular systems. And yeah, did that answer your question? What was the last one? $2 to $3 million.
We'll go right here.
Hi, thank you so much for your presentation. You did a great job making something very complicated, really widely applicable, so thank you for doing that well. I just had a question about your concerns about the ability to scale this to the general population, like thinking about all the constraints post-studies.
Yeah, so it might be helpful to say there are a couple of different approaches to phage therapy. One is a very personalized approach where you actually get the bacteria sample from the patient, and then you look at your bank and take your best set of phages directly on that sample. Then you manufacture that and you take that back to the patient and treat them. And that's how a lot of phage therapy is done. A lot of the emergency INDs that have been done and have been successful have been that way. We decided to take a different approach, which was to invest in a lot of this upfront. To be able to get what we refer to as a fixed phage cocktail, which is something that we could give to many patients. So we have the manufacturing capability to be able to scale this to commercial to treat patients throughout the United States. The particular product that I mentioned is for E. coli in urinary tract infections. So that is maybe a different level of scale than all types of infections and all types of bacteria. But we're also working on other types of bacteria as well as other indications to kind of spread out from our initial lead product.
Okay, and for the final question of the night, we'll go over here.
So this is a biology question. Can phage therapy actually clear an infection, or is it kind of like biocontrol of invasive whatever, where you just keep the level down to the point where you're no longer symptomatic?
Yeah, wonderful question, and the short answer is we don't know quite yet. But I think that there are a couple of options, both of which are positive for patients. One is what you mentioned, where you keep it to a level that's asymptomatic for the patient. We obviously would like to get to a point, particularly for urinary tract infections, where you can completely clear that. In our initial study that was non-placebo controlled, most of the patients had undetectable levels of E. coli out to 34 days, so they could come up after that. We don't know for sure. The other option is that phage can also make bacteria resusceptible to antibiotics that they were not previously susceptible to, and they also can bind to virulence factors or other ways that bacteria use to affect human cells that they can adhere to or invade to human cells. There are phage that target the mechanisms essentially that the bacteria uses to do that. So you could imagine a scenario where you push evolutionarily a bacteria to become what was once a pathogen to something that's a commensal that can live in harmony.
Thank you, Dr. Penke, everybody.
Thank you.